Thank you everyone. Welcome to join us for today's molecular connectivity online series. My name is Cindy from New Jersey Institute of Technology. In case you are new here, our molecular connectivity working group is a group of scientists that aim to promote molecular imaging as a tool to study brain connectivity. We organized this online series to promote scientific research and discussions on molecular connectivity. If you are interested in the work of our group, please go to our website and follow our newsletter and Twitter and LinkedIn. So today, I'm pleased to have Mr. H.Deery. Mr. H.Deery is a PhD candidate in Monash University from Cognitive Neuroimaging Lab, led by Professor Shama Jamadar. His research interests are in understanding brain functions and cognitions across the adult lifespan, including unique and potential shared pathways in aging and lifestyle diseases. His work includes the use of simultaneous functional PET and MRI to investigate the impact of insulin resistance on cerebral glucose metabolism and cognitions in mid and late life. Today, he will talk about molecular connectivity in aging. After the talk, I also invited Ms. Chunmeng Tang that will moderate the discussion session. She is currently a PhD candidate in KU Leuven in Belgium. Her research focuses on advanced brain PET image analysis through machine learning in quantification, classification, and connectivity analysis. Once again, thank you for coming, and let's welcome Amish. Thank you.
Wonderful. Well, thank you very much for the introduction, Jin, and thanks for the opportunity to be here and share our work today. Before I start, I just wanted to acknowledge the broader team that's been involved in this work, particularly Shana, who's my supervisor and whose lab I'm part of here at Monash, and she developed much of the methods that I'll talk about today. I also just wanted to mention that this work was recently accepted for publication in Communications Biology. So the paper is being copy edited at the moment, so should be online available quite soon. So by way of background, I just wanted to talk a little bit about why we're interested in studying aging. And it's estimated that the global population of adults aged over 60 years will almost double, at least from several years back, being 2.1 billion. And while aging is not a problem in and of itself, it is a major risk factor for cognitive decline. We know that extensive brain changes occur in aging, from the molecular level to the functional level. And older adults typically show a decline in a range of cognitive domains or functions, which include executive function and attention. So these are things like the ability to think abstractly and problem solve. Although cognitive decline is considered typical or quite normal in aging, it's not inevitable. We know that some older adults show clear signs of decline by the age of 60, whereas others can retain excellent cognitive function well into their 80s and beyond. And so this really raises the question that we're addressing in our research in terms of what can be done to optimize cognitive function through aging. And to help address this question, we're using molecular and functional imaging to study connectivity changes in the aging brain, with the potential for it to serve as a biomarker for cognitive decline. One thing I wanted to mention up front is that we define connectivity as the temporal dependency between spatially remote neurophysiological events. And I wanted to mention that because this definition is important in the context of metabolic connectivity that I'm going to be discussing throughout the session, because we're using a time series of glucose metabolism within individuals to look at regional coherence of the time series, which distinguishes this approach from other methods that have been used in the past, such as a cross-subject covariance. And I'll be talking a bit more about this as we go through the session because it is an important distinction. And finally, I just wanted to mention that In my PhD, as Jin mentioned, I'm quite interested in understanding how health status, such as insulin resistance, moderates metabolic connectivity. But today I'm going to be focused on our first question here, which is around aging. In terms of a bit more background to the approach that we're taking, I wanted to discuss a bit more about the molecular imaging methods that we're using. The method reflects the fact that the brain is, when the brain's active, it's accompanied by an increase in blood flow, oxygen use, and glucose uptake to the local area to meet the energy demands of the neurons. And the change in blood oxygenation underpins the bold signal in fMRI. And of course, fMRI has been widely used to study connectivity in the brain and has revealed much about the functional organization of the brain. But it does have its limitations, and perhaps the most important in the context of our work is that the components of neurovascular coupling are differentially impacted in aging. So this makes the basis for understanding functional changes quite difficult to interpret in aging. In comparison to this, glucose metabolism, as measured in PET, has the advantage of being a quantifiable and direct measure of neuronal metabolic activity, primarily reflecting activity at the excitatory synapse. It also does have its limitations as well. From a connectivity perspective, PET has, particularly historically, been limited by poor temporal resolution. So PET methods often in the past have involved administering the tracer as a bolus, waiting, say, 20, 30 minutes for that tracer to accumulate in the brain tissue, and then capturing a single image, like you see in the bottom left of the screen here. Now, this can be powerful in terms of using like kinetic modeling to estimate the rates of glucose metabolism in regions, but it doesn't create that time series that allows for the connectivity analysis. For that, we need to create multiple images. They need to be generated to create the time series. And so the analysis then comes akin to what we do see in other modalities like fMRI and EEG. And that's what you see in the bottom right image here on the screen. With a glucose time series, we can then study the brain as a network using connectivity analysis or graph theory metrics. And we can index the topological patterns and the efficiency of information transfer across the brain. And so that's really the type of analysis and method that we're interested in this work. Over the last decade or so, a lot of progress has been made in these fPET methods to allow this time series to be analyzed, and increasingly with faster temporal resolutions. So a landmark study by Villian and colleagues back in 2014 showed that glucose signals in task-relevant visual regions could be identified at a resolution of 1 minute temporally when viewing a flashing checkerboard. And since then, a number of authors, particularly the group in Vienna, has published papers reducing the frame duration to 30 seconds, and even now pushing spatial resolutions of less than 10 seconds. In our group, again led by Shana, we've examined whether combining a bolus administration with a constant infusion can actually create a better signal-to-noise ratio across the duration of the scan compared to a bolus alone or infusion alone. And Shana's study has published some data here showing that with this method and a 16-second frame duration, we can, in fact, identify a strong signal in the task-relevant visual regions when viewing the checkerboard. So you see that in the bottom left image on the screen. Just the other thing to highlight here is I mentioned the importance of distinguishing between a cross-subject covariance to study connectivity and creating individual time series, what we're calling fPET here. And you see that, again, Shana's published some validation work showing that, in fact, they do identify different aspects of brain connectivity, so further supporting the importance of looking at within individual time series. And then on the right-hand side here, also showing that there's similar but complementary patterns of connectivity between fPET and fMRI. Finally, I just wanted to mention on the right another publication in the same data set looking at the connection or correlation association between metabolic connectivity and cognition and fMRI and cognition. And again, importantly, the team found similar but slightly different mapping to cognition, particularly executive function. So further supporting the idea that studying glucose metabolism in the brain will yield additional insights beyond those of functional connectivity. Before I move to start to share a bit about our current study, I thought it's worth just pausing and talking a little bit more about brain connectivity and network measures, and then share what we know so far from the research or the literature on what happens to connectivity and network measures in aging. No doubt many of you on the call are familiar with the concepts of establishing and measuring networks in the brain. And over the last 2 decades, this has often been done treating the brain using concepts like small world networks and graph theory. Now, in the interest of time, I'll focus on the metrics shown to be important in aging and those that are the focus of our research or the study that I'll share today. So typically, we're looking at nodes within the network indicated by the signals, the open circles in the graph or figure on the right. The edges between the nodes are shown by the straight lines. And by looking at the connections between the nodes, we can make different measures of the effectiveness of information transfer within the brain. So, for example, we have a measure we call global efficiency, which looks at the number of paths or the average of the distance between nodes and all other nodes across the brain. And this is also an index of global integration. So the more efficient the signals, the more integrated the brain is considered to be. Local efficiency is a similar measure, but it's looking at a neighboring subgraph. So it's looking locally at neighboring nodes, and it's telling us about information transfer more locally. There's a number of different centrality measures we're using between the centrality here, which is essentially the proportion of time that the node spends as part of a shortest path. So the more time it spends, the more central it is to communication to other regions. And then finally, degree is a measure of the number of edges from that node to all other nodes. And this indicates the degree of connectedness of that region within the network. So I'll be sharing with you results on these 4 measures from our data set. But I did also just want to talk briefly about the importance of hub regions, because our analysis will focus on metabolic hubs. And there's an organizing principle of the human connectome that there's a trade-off between minimizing energy cost and maximizing communication efficiency. So it's been established, again, particularly with fMRI, that cost minimization constrains the connections to local nodes in the network, so more like the local neighborhoods that I was talking about. And that creates a network structure that's modular with high local clustering and efficiency. However, it's also recognized that cost minimization principles are insufficient to describe the network and its properties. And so there are these hubs, which in the figure here are shown in red, that are particularly important to network communication because they have long-range connections that allow information to be transferred across the brain. The other thing we know about hubs is that they have what's called a high wiring cost or a wiring cost premium, which means they're using a large amount of metabolic substrate to maintain these connections. And although they have this high cost, as I mentioned, they also have very high value to information transfer. So we'll return to the concept of hubs when I share results with you from our study. I mentioned I was going to briefly share, well, what do we know so far about brain and metabolic network changes in aging? What I'm sharing on the screen here is 3 recent systematic reviews that we've undertaken. The first was on large-scale resting state networks in fMRI, 145 studies, and essentially we found that there's lower within-network connectivity and higher between-network connectivity in aging, which drives a structure that is less modular and more integrated globally. And we also found that there was less efficiency So the signals need to cross more of those paths that we talked about in the networks. We found the inflection point for many of these changes is around the 4th or 5th decade of life. Now, the reason I'm highlighting fMRI here is because there just isn't the same body of research in fPET. So we're using the fMRI literature to help establish some of our hypotheses that we want to test in this research. Then on the right-hand side, we used, we analysed PET research in these systematic reviews, and we also undertook 2 types of meta-analysis. First, pooled effect size meta-analysis, which is on the left-hand side. And we did find that there are significant reductions in metabolism, cerebral glucose metabolism across the brain, particularly in the frontal lobe. Now, those reductions are reduced when you take into account that older adults also have a reduction in cortical thickness, so they're losing metabolic tissue. But nonetheless, even after adjusting for those differences, particularly in the frontal lobe, the differences were quite strong at 11% reduction. The second meta-analysis was an ALE analysis. We identified a number of different clusters of reduction in glucose metabolism. The 2 largest were in the frontal and temporal lobes. And so collectively across these meta-analyses, we're really concluding that there are significant reductions, but particularly in the frontal and temporal regions of the brain in aging. And again, we're using that as some of to drive some of our hypotheses in our research on connectivity. So to the current study, 2 primary goals with this particular data set and analysis that I'll be talking about. The first was to investigate the alterations in the metabolic connections in aging and their relationship to cognition. And the second was to measure the cost of the metabolic networks using the rates of glucose, so indexed across the entire scan in hub regions, and to contrast that relative to the role that the hubs play in information transfer across the brain. So here we're indexing the cost of maintaining those metabolic networks. We had 5 hypotheses. The first was that age differences in metabolic connectivity will be predominantly frontal and temporal. So again, derived from the fMRI research and the meta-analyses on glucose metabolism. Secondly, that metabolic connectomes will show lower within and higher between region network connectivity amongst older adults, again, derived from the fMRI literature. Thirdly, that the metabolic hubs of older adults will show lower efficiency, degree, and centrality than those of younger adults. Fourth hypothesis is related to the concept of the cost of maintaining the network, and we hypothesize that there'll be higher cost to the hubs than the non-hub regions, and also that there will be a greater proportion of brain glucose metabolism used to support network connections in older than younger adults. And then finally, that the graph metrics that we're measuring for the metabolic networks will be associated with cognitive performance. So in terms of methods, we recruited 40 younger and 46 older adults from the local community. The mean age of the younger group was 29, the range 20 to 42. And for the older adults, the mean age was 76 and a range of 60 to 89 years. I just note that we collect a number of other demographics, things like BMI, education, level of insulin sensitivity. And we found across the sample that those were in the normative range for age-relevant Australian populations, so based on data that the Australian government publishes. So we have reasonably representative samples of younger and older adults in Australia, in the Australian population. Participants underwent 3 testing sessions. The first was an online demographic questionnaire. They then came to the lab, underwent a cognitive battery that included memory, both episodic and digital, so both episodic and working memory. So episodic memory we indexed via the HVLT. Working memory was a forward and backward digit span task. And then we also had 3 computer-based tasks, task switching, which indexes cognitive control, a response inhibition task for inhibitory control, and digit substitution task for visuospatial performance. With a couple of those measures, we're also measuring reaction time or processing speed. So I'll share results for that as well. Reason we selected those domains is they're, again, well established to show age-related declines, so well validated for studies in aging. In the third session, participants underwent a simultaneous functional PET and MR. I've got the scan parameters and pre-processing steps here on the right, just to touch on or highlight a couple of the key ones. As I mentioned, we found by administering the radio tracer as both a bolus and constant infusion, we get that strong signal to noise over the period of the scan. So we had administered half as a bolus and half at a constant rate of infusion over 50 minutes. And then really just to mention, we're using well-established reconstruction and algorithms to pre-prepare or pre-process the PET data, including things like motion correction. So if anyone's interested in more of the pre-processing steps, very happy to share that. A summary of the analysis workflow or pipeline. So starting here at the top, the individual fPET images after pre-processing were loaded into the CON toolbox, which is the MATLAB toolbox. They underwent denoising, which is regressing out the white matter and CSF signals. And then post that, each individual subject's data went through a series of steps. We parcellated using an anatomical atlas here, the Harvard-Oxford Atlas. I should mention that it's not yet well established whether functional PET data is well reflected in more established fMRI parcellations, but we have in our supplement to our paper repeated the analysis with the Schaefer Atlas. And if anyone's interested, again, I can talk a little bit about that later. So in CON, we established a time series. We calculate the individual correlation matrices across the regions. We also then calculate the graph metrics after establishing an adjacency matrix. So those graph metrics were global and local efficiency, centrality, and degree, as I mentioned earlier. If we drop down to the bottom here, we did index the cerebral metabolic rates of glucose across the entire scan. Again, parcellating for the Harvard-Oxford Atlas. We were using PATLAC graph analysis here. And then importantly, the middle panel is the summary of the analysis that I plan to share now. So we're going to look at the connectomes, the group average connectomes. We're going to look at the features and functions of the metabolic hub regions, the graph metrics, and the relationships to cognition. I thought it was just worth quickly showing you time series for an individual participant from 2 regions. Reason for doing this is with PET, one of the challenges in the analysis can be the impact of that initial baseline uptake of the radio tracer, particularly it creates quite a steep ramp in the initial period. So this is really just to show that the pre-processing and denoising steps that we're taking are doing a good job of establishing a signal with variability around a relatively stable baseline. So some interesting results here. Firstly, the younger and adult connectomes. And the most salient characteristic for both groups is that the older adults and the younger adults have high connectivity strength for regions in the frontal, motor, and parietal cortices, both within and across hemispheres. So you really see that in these darker red colors in the matrices. Medial connectivity was also quite strong to the frontal, motor, and parietal regions, and again for both younger and older adults. In contrast, the between-region connectivity was relatively low for the limbic, temporal cortices, and subcortex for both the younger and the older adults. And then a second salient feature that sort of jumps out here is that the connection strength looks to be higher in the younger than the older adults. So to further test and quantify that, we calculated within and between region connectivity by averaging the nodes within the regions. And you see the younger and older adult matrices on the left here. And then we undertook statistical tests to establish where there are significant differences between the two groups, and they've been FDR corrected. And what we see is that, again, these stronger connections within the frontal regions for the younger relative to the older adults. So the red indicates a stronger connection for younger relative to older. Strong connectivity from the frontal region to the motor and parietal regions, particularly in the right hemisphere, strong connections from the temporal across a number of the other, particularly associative areas in the brain. And just finally, an absence of subcortical connectivity was also of note. So for us, this really extends the research I shared earlier, showing that cerebral metabolic rates of glucose decrease in aging, particularly in the frontal and temporal regions. So we did find strong support for our hypothesis, particularly in terms of frontal, slightly less so in terms of temporal, but nonetheless quite consistent with the previous research using static PET. Moving to talk about the metabolic hubs. So firstly, just on definition, in the fMRI research, I talked earlier about wiring cost. It's been found that the wiring cost of connectivity increases monotonically with nodal degree, so the number of connections from a region or a node to other nodes, which makes sense. The more connections, the higher the cost of maintaining those connections. It's also been found with that cost, that trade-off between cost and integration of signals across the brain, that is optimized somewhere in the range between the top 5 and 35 of relative edge strength. And so we used, again, this to shape where we thresholded our matrices and calculated the graph metrics, and we landed on both top 10 and top 30% of network edges, and repeated the analysis of each of those levels of thresholding. To establish the metabolic hubs, we simply calculated the average on the measures of centrality, efficiency, and degree, and took regions that on at least 2 of those measures were more than 1 standard deviation above the mean. And that's what you see in this chart on the left-hand side. Those shaded red are simply the regions that are above 1 standard deviation. And with that approach, or I should say the reason we adopted that approach is there really is no single agreed definition of what constitutes a hub. All of these metrics have been used in the literature to define hubs previously. So by adopting this approach, we look to try to ensure that we at least covered these key features of hubs that have been established previously. With that definition, we found 13 hubs at the top 10% of network connections. And they were in the frontal poles, the middle superior frontal gyrus. So these are in the frontal areas of the brain, in the motor areas, the precentral and postcentral gyrus, the lateral occipital cortex and occipital poles, the cingulate gyrus and precuneus. And then interestingly, when we ran the analysis of the top 30% of edges, so including more connections in the network, we found 2 additional regions met the definition for hubs in the frontal gyrus and the frontal orbital cortex. The first thing that we wanted to establish here was whether the hubs do in fact have this wiring cost premium. Are their rates of glucose higher than non-hubs? And our analysis supported this hypothesis. We found on average just over a 4% higher rate of glucose metabolism in the hubs, the 13 hubs and the 93 non-hubs. And these are individual participant differences between the hubs and non-hubs here in percentage terms. And you can see that for 83 of the 86 participants, there was higher metabolism in the hubs than the non-hubs. So again, supporting the idea that these hubs are particularly important, not only for efficiency, but they do have this high wiring cost premium. What I'm sharing next are the age differences in efficiency, centrality, and degree of the hubs. So what's quite interesting here is that when we look at the top 10% of edges, we found significant age differences in posterior regions of the brain, where older adults had higher global efficiency. So these hub regions are more connected to the rest of the brain in older adults. However, what's interesting is that when we introduce more connections into the network, so here at the top 30% of edges, those posterior regions no longer show age differences. And in fact, the direction of the age differences reverse and are evident in the frontal regions of the brain. So this pattern was also found with betweenness centrality, so indicating that these hubs are central in communication at these network thresholds, whereas degree, you can see, tended to maintain the consistent advantage for the younger relative to older adults. So this suggests that posterior nodes are not as central in global communication in older adults in a denser network, because the additional connections dilute their ability to maintain those stronger connections or integrations across the network. But in contrast, for younger adults, even in where more network connections are considered, the frontal hubs show robust and more meaningful links between the nodes. Because these nodes have high global integration at this level, it suggests they're highly central and efficient in facilitating communication across the brain. And as I'll talk about in a moment, these patterns also had important implications for cognitive performance of the younger and the older adults. Also showing the results here for local efficiency. When we look at local efficiency, younger adults have higher efficiency than older adults in hub regions across the brain. And that's true at both the top 10 and top 30% of network edges. The largest differences are, again, in the frontal poles, the middle frontal and superior frontal gyrus, so the frontal areas of the brain, but also in the precentral gyrus and the lateral occipital cortex. I think the take home from this particular analysis is that these results suggest local efficiency of hubs in younger adults helps to minimize the cost of maintaining network communication via fewer paths to other connected regions, particularly in those local neighborhoods. We also calculated an index that we called the glucose cost index. So this is the ratio of the metabolic rate of glucose indexed across the scan in the hub relative to the graph metrics for each of the hubs. And so the glucose cost values reflect the metabolic cost of maintaining the network communication as measured by the graph metrics. I'm not sharing the data here today, but we have shown that there's lower metabolic rates of glucose across the brain for older compared to younger adults in this sample, including in the hub regions. And so this glucose cost index, if we take the example of global efficiency across the top here, is suggesting that a high glucose cost is evident for global efficiency, reflecting the fact that hubs that have lower available glucose to support more paths between regions in older adults. So the reason I say that is the red, again, is indicating that this measure or this glucose cost index is higher in older than younger adults. So the takeaway is that older adults use a larger proportion of a smaller glucose budget to support integration of hubs across the brain, which they also do less efficiently than younger adults, as we'd shown on the previous slide with the global efficiency metrics. The last series of analysis I wanted to share then is looking at the associations to cognition. And for this, we used canonical correlation analysis to identify the linear combinations of the graph metrics in the hubs and the cognitive variables that most correlate with each other. And so with canonical correlations, the highest number of variates that can be identified reflects the smallest number of variables in the data set. So in this case, it's 10. We have 10 cognitive domains, including the measures of reaction time and processing speed. For global efficiency, we found one significant variant, so meaning that the hub regions, the measures of global efficiency, loaded onto one single variant over here, and similarly for cognition over here. And so at the top 10% of edges, we're seeing at the top here, that the lateral occipital lobe and the precuneus global efficiency in particular have the strongest loading on the efficiency variant and is most strongly associated with lower episodic memory, visuospatial processing, cognitive control, response inhibition, and slower reaction time. Interestingly, when we look at the top 30% of edges, the direction of the loading of the efficiency reverses, so in line with what we saw with the graph theory metrics. So here, lower efficiency in the frontal regions of the brain in particular are associated with worse cognitive performance. So you recall that this essentially reflects the profile that we saw in younger and older adults, with younger adults having much stronger global efficiency at the top 30% of edges, and older adults having stronger global efficiency, particularly in the occipital areas of the brain, the top 10% of network edges. Here's the analysis for local efficiency, and we found that lower local efficiency at both the top 10 and 30% of edges across many of the hubs was associated with worse cognitive performance on most domains, most notably, again, episodic memory, visuospatial processing, cognitive control, reaction time and processing speed. And so this is suggesting that hubs are key players in network communication and are prone to age-related alterations that impair cognitive performance. So just to conclude, 5 key findings or takeaways from the study. Firstly, that aging is characterized by an altered metabolic connectome, and we see reduced capacity of hubs to connect to other regions, particularly hubs in the frontal regions in aging. Secondly, that hubs have high value in terms of their information integration across the brain, but they also have this high wiring cost premium in terms of the amount of glucose metabolism that they use. Younger adults use a high proportion of the available glucose in the hubs, in the frontal regions in particular, which support that information transfer across the brain. We saw that with particularly the global efficiency and between the centrality amongst the younger adults in the frontal regions of the brain. In contrast, older adults have a higher glucose cost of their cognition, so they're using a higher proportion of that smaller energy budget. They're using it less efficiently in terms of the glucose metabolism is less efficient, but also those connections, particularly between the posterior and medial hub regions. And this pattern is associated with worse cognitive performance. So finally, I think good news for the field, our results highlighting the fundamental role that metabolism plays in supporting information transfer in the brain and the unique insights that metabolic connectivity provides into the aging brain. So with that, thank you for listening to the session, and I guess we open up for questions and discussion.
Yeah, thanks for the nice presentation. I really enjoyed the talk, and it's also very interesting to me. So we have a question from Xin.
It's just applause. Okay, so yeah, thank you for the talk. I do have a question actually. So, so you mentioned that there may be some differences of connectivity if you measure it using fMRI and PET. But today the results you show are mostly PET, right? So have you compared the same analysis and whether the results are different or the same?
Yes, we have, and actually it's available in another preprint. So we contrasted using similar measures, the fPET and fMRI, and found, I think, two key things. One is that the fPET is better able to predict age and cognition than fMRI, which is, I think, quite interesting given the strength of the fMRI literature. And secondly, that particularly the between region connectivity pattern is opposite in fMRI. So we do tend to see stronger between network connections in older adults in fMRI, whereas, as I'd shared today, we're seeing much stronger between network connections in the fPET.
So if any differences you observed between fMRI connectivity and PET connectivity, how do you interpret, like what are the mechanisms that may cause this? Do you have any insight?
Yeah, that's a great question. I think it gets back to what PET and fMRI are measuring in terms of neuronal metabolism and how they're indexing that. So the fPET is directly measuring glucose metabolism at the synapse, as we talked about in the introduction, whereas the fMRI bulb signal is influenced by a range of different factors, particularly in the neurovascular coupling. So it can be oxygen extraction, it can be blood flow, and these things are differentially changing in aging. So while we can't be certain of the exact mechanisms, I think what it is saying is that, importantly, fPET and fMRI are indexing different processes in the brain, and we're able to identify those differences, through the signals. But we think it's probably in the way that the fMRI is measured.
Go ahead.
Sorry, my Zoom somehow doesn't work. Can you guys hear me? Sorry, I came in a little late. I do have one just methodological question and then one more broad mechanistic question that might be somewhat speculative. But I like the canonical correlation analysis. Maybe you explained it before. The one thing I was confused about is your global efficiency measures still seem to be local because they are focused on different anatomical structures. Was there something that I didn't get, or I thought global efficiency would be a global measure where, you'd somehow take the inverse of the path, the shortest path length between all possible region pairs.
That's exactly right. So that's what we're doing in calculating global efficiency. It's from the hub to all other connections, possible connections in the brain. Yeah.
Okay, that's why it's a kind of local measure, because it all terminates on one hub.
Correct. Yes. Whereas the local efficiency is from that hub to its near neighbors.
Okay. All right. Actually, that's great. That makes sense. Yeah, I really like the metabolic cost argument that you made, and I wonder, and maybe this partly speculative. Do you think this would be partly mediated already, something by structural atrophy, or maybe you haven't measured this, or is it really unique phenomenon that maybe precedes structural atrophy and is maybe driven by things like mitochondrial dysfunction or inflammaging or some of these other things? I don't know whether your blood measures. Just your speculation would be interesting.
It's a great question, one I'm very interested in, Christian. So in this sample, we've looked at just the metabolic rates of glucose in the same regions of the brain. We do find large reductions in aging that are moderated by cortical thickness or loss of tissue, as you say. So we do think there'll be a moderating factor involved. In terms of mechanisms, as you say, speculative, but broader field has shown that the efficiency of global metabolism does reduce due to things that you mentioned, like mitochondrial function. Increased insulin resistance is something I'm quite interested in. And so one of the next planned analysis look at the role of insulin sensitivity on some of these processes as well. So I think many of those things will be at play in the aging brain as well. So keen to try to tease out shared mechanisms in sort of aging and health.
Do we have any other questions?
I have a question. I just wonder, like, sometimes I think especially from people in the pet world, we get asked if, is it worth it to go into these rather complicated metrics, right? Why should we do connectivity if we can just look at metabolism? So do you have any results as to whether metabolic connectivity, for example, predicts cognition better than just looking at glucose metabolism? Or what do you expect if you didn't check it?
Yeah, great question, Ariana. I think we often get two questions related to that, don't we? It's like, well, what's the utility of PET over fMRI? Particularly given the radio traces that are involved. But to your question, we are actually now working on some analyses looking at that exact question. Is there a direct impact between the rates of glucose and cognition, or is that effect indirect via the variability in the glucose signal, the glucodynamics? Our preliminary analysis would suggest that it's via the glucodynamics, which would make sense because that's what's really establishing these emergent properties in the networks, and that network architecture then is linked to cognition. So we want to continue with that, exploring that question, but it's quite promising. And in terms of my other interest, we're also finding that the more insulin resistant somebody is, so our sample is largely healthy, they're not clinically insulin resistant, but we can still assess it on a spectrum, that is driving the answer to your question as well. It's more impacting the coherence of the signals than it is the rate of metabolism. So there seems to be a causal chain through to cognition by the variability in the signal that we're indexing through connectivity analysis.
Okay, yeah, it's well, I guess aging is a very complicated object of study. I have the impression studying Alzheimer's or like neurological conditions is much easier than aging. Like,
yeah, one of Shana's visions for this work was to include at least an MCI group. So maybe something for the future. Yeah, it'd be really interesting to see how these metabolic connectivity measures are different, if at all, in those populations.
Hi, Jimmy. There's a question in the chat.
Yeah, I saw. Okay, so we got a question from the chat. Thanks for the really great talk. Have you or other have tested the retest repeatability, or I think it's reproducibility of molecular connectivity measures?
I haven't personally, but Shana has done some of that work in the lab, unpublished data. But it's a good question. I think it's something that we're going to have to increasingly look at in the field as we establish this work more. So yeah, I think the answer is I'm not aware that anything's been published yet, but something that needs to be done.
If I may follow up, like, since we're going a bit into methodological questions, like, I don't know much about constant infusion PET, but like, how, let's say, established are the preprocessing pipelines? Like, is there like already some kind of best methodological practices that you would recommend? Is there also any testing in terms of the robustness of your results compared to different analytical pipelines? What's the status of the field? Because of course it's quite new compared to the old style static PET with bolus injection, right?
Yeah. Again, I think this is a really important direction for us to consider as a field, relative to the well-established pipelines in fMRI. We're really at baseline level here. I think with the work we're doing, some other groups as well, some that I cited in the introduction, I think we're making good progress, particularly in aspects like looking at, do we need to smooth not only the spatial but also the temporal signal, how to deal with that baseline issue that I mentioned. So we're not yet to standards, Ariana, but I think we're making progress. And I think, given the analytics now that are available, it's only going to improve our ability to pre-process this sort of data.
Yes, just add a little bit. I think Julian Chen from MGH, they are also analyzing the same data of your data, right? And I saw there's a new preprint there, the thing that like removing some global trend may have the impact of the results, something like that. There's a new preprint.
Right. Yeah. So when like using data-driven approaches to identify the networks, it's often the global signal is removed, or even the signals are Z-scored. So I think if I'm thinking about the same preprint as you, Jin, they're talking about taking some of those steps. Yeah.
Yeah, any other questions?
Tommaso, if you are here. Because no, I mean, you don't have to answer, but like, I was a bit wondering, like, since we are discussing, should we do fMRI, should we do constant infusion PET, should we do PET or connectivity, or just look at metabolism? The other question is, should we really do constant infusion, or can we just, do a bolus injection and just do a standard dynamic scan, which is a bit more accessible, maybe than a constant infusion scan? So I know that there's been some methods that have been proposed, right, to analyze the time activity curve in standard dynamic scans. So you see like any advantage in still choosing a constant infusion protocol, or you think maybe the two methods could be relatively equivalent, or maybe not? What is your opinion of both, if I can get you?
Hi, Ariane. Sure. No. So the main advantage of having the constant infusion relies on the at least potential capability to evaluate resting state fluctuations, right? So in the way that Amish and Sharna and the people from the Vienna group and people from NGH and others have tried to do. So you're trying to remove the baseline and you evaluate those resting state fluctuations under the assumption that they reflect neural activity, which of course is a big if. It would need to be demonstrated, because I mean my hunch is that they probably represent a combination. I mean it's very, very, very likely that they represent a combination of neural activity and measurement error. So simply variance related to the noise related to the measurement process and the sensitivity of the scanner that you're using. So with a constant infusion, you can definitely try to access that aspect with the bolus plus constant infusion. With a bolus injection, instead, you cannot really do that, because you never reach any kind of steady state. And so, I mean, for sure, you could try to do the same. So you could remove the baseline, you could remove the whole brain average curve and leave only the fluctuations around it, and try to evaluate the metabolic connectivity on that for bolus injection too, which we have done in the past. We mainly with FDG PET, and so we have not. I mean, in the end, we have kind of preferred to go towards a more kinetic based approach, which is the one that we've chose to use, which evaluates the distance between the time activity curve using Euclidean distance. And so in that case, you're evaluating how similar the amplitudes and the overall kinetics of different time activity curves are. But of course, in that case, you are losing, I mean, you're not really losing, but you're not considering very much the fluctuations. You are mainly driving your similarity, and so your connectivity measures based on, again, the larger shape, the overall dynamics of your time activity curve. So it's kind of a different way to look at it. So yeah, so that's kind of the answer. So for sure, our approach can be done with either a bolus injection or with a constant infusion. It's kind of the same because it's kinetics, so you can assess those similarities irrespective of the protocol that is applied. Instead, the resting state fluctuations, it makes a lot more sense to do it like Sharna is doing and the other groups are doing, so to try to reach some sort of steady state and just to try to isolate those signal fluctuations. Sorry for this very convoluted answer, but yeah.
What do you think, Amish? Is it so? I don't know. I guess constant infusion is a bit more direct. I don't know how to say. Or last.
Yeah, I agree.
Less assumption, it seems, right? I mean, you're not removing any phasic signal. But by the way, I think this is all super interesting. I mean, we're running out of time. But you know, I really think a work group could maybe once have some event about, I mean, all of these issues. I wasn't aware that there's so many hyperparameters even in the application of the PET, let alone the analysis, which I think, I'm sure there are lots of motion artifacts and lots of other things, and whether you take binarized connectomes or weighted connect. I mean, this would really be a topic, whole meeting almost, or maybe virtual meeting.
Yeah, I mean, building on Tommaso's point too, one of our aspirations is to be able to quantify the signal using kinetic modeling, because I think that will help address some of the concerns about like baseline uptake and its impact through the connectivity analysis. But that does require, I think, a next level of analysis, particularly to be able to quantify the signal, maybe in a sliding window type approach. So something again, maybe for the future, Ariana, and to your point about sort of standards and the quantified signal, I think would be something that then is more comparable across studies. We should get a rate of glucose in the time series.
We also got another question from the chat. What do you think would potential influence has a visual stimulation, like walking a helicopter flight over Hawaii, on the data in that regard?
Yeah, good question. I mean, it's a methodological consideration we had in designing the study. It's quite a long scan. We had participants are in the scanner for 90 minutes, and so we had some concern that pure resting state scan, they may fall asleep. So we decided to go with something that's naturalistic, which does make, I think, interpretation of the signals a little bit more challenging. So again, I think that extending this into task-based analysis and pure resting state would be worthwhile.
All right, thank you for all the discussions, and thank you again, Amish, for the talk, wonderful talk. So if we do not have further questions, thank you for all coming. So that's it for today. And we do not have the MCOS for December because that will be holidays, and then we will have next sessions on January. Okay. Thank you all. Sarah, what is?
Just the link to register for the next MCOS if you want already, to not forget about it. It's again about aging, but a bit more, I think, biologically more hardcore with epigenetic changes.
Right. Yes. Okay. Thank you for joining today. Thank you.
Thank you.
Thank you very much. Good night.
Good,
Hi.
good night.
Good afternoon.
Hey.